Hi, and welcome back to the Slug Show. This is episode 6. Why am I in my pajamas?
Is it A, because I am a tired, overworked grad student who doesn't have enough time to do laundry? B, I'm a tired, overworked grad student who doesn't care about what she wears to work anymore because the only living creatures that see me are sea slugs? C, I'm spending my night at the museum? Or D, all of the above? If you guessed D, you are correct. I've made a little tradition out of wearing my pajamas here when I'm staying the night. So why am I staying overnight? It's definitely not because I love my slugs so much and I just can't live for a single moment without them being here. So I'm staying the night tonight because of research. One of the main things I'm trying to study with this project is what exactly happens to a starved slug when it gets something to eat. What happens inside its digestive system to facilitate either incorporating chloroplasts, hanging onto them for later so they can do photosynthesis and maybe make some energy for the slug, or in a super starved animal that doesn't have any energy right now, what exactly makes them digest that chloroplast immediately because they need the energy so badly. When these animals are well fed and completely packed with chloroplasts, they can withstand months of starvation. But when they've been starving and they get food again, they don't save anything for later. They digest everything they get, including the chloroplasts, and they use that energy from this digestion to rebuild their bodies. Our human bodies do the exact same thing. When we are starving, our bodies first consume any extra fat we have. After that is gone, our bodies will digest anything that is less important than our vital organs, mostly our muscles. Finally, the body can't help but digest our most important muscle, our heart. And that's why severe anorexics have a really high chance of heart failure. It works the same way in slugs. A starving slug eats away its own tissues to preserve what matters most. They preserve the circulatory system, the nervous system, the genital system, because, well, these are short-lived animals. They only get one chance at reproduction, so they're not going to eat that away. In these pictures, you can see a deteriorated animal who's suffered severe weight loss compared to a normal, healthy animal. When I give this starved animal food again, it's not going to start building up a pantry full of chloroplasts, at least not immediately. We know that it takes them about a week to rebuild themselves before they start sequestering chloroplasts again. That means a week of constant digestion. It also means that they change their ability to hang on to chloroplasts depending on how hungry they are, how much energy they need at that moment. And this means that the ability to hang on to chloroplasts comes from the slug itself, not just the chloroplast. And I want to know how they do this. In some of my experiments, I starve the animals down until I see that their PAM values, those values that tell me if photosynthesis is still occurring, are pretty low, say 30 to 40%. In Elysia timida, that's after a couple months. When a slug hasn't eaten for a few months, it is super hungry. In my experiments, I give it food so I can see what happens right after it gets this meal. Now, I am looking at this process on a cellular level. That means what individual digestive gland cells and individual chloroplasts are doing at the moment the chloroplast enters the digestive gland cell. And I want to know how that's different in animals that need to eat everything and digest everything right now because they're starving, or, and how it's different from animals that have been eating pretty consistently. They're not super starved yet, or they've been eating the whole time, and they can afford to take that chloroplast and add it to their pantry. They don't need to digest it now. I want to see how these two are different. Examining cellular processes is really tough because they occur on such a small scale, and they occur over time. I can see some of these things through traditional light microscope, like the one you see right there, if I slice it first. But some things that I'm looking for are too small even for that. That microscope there has a 100-time objective lens, and that's still not good enough for some of the stuff I want to look at. To see individual membranes in the insides of organelles, we need to go much deeper. We need much stronger equipment. Enter the transmission electron microscope. These machines are huge. Think whole room huge, and whole room next to it to cool them down huge. And these machines can allow us to get to the level of detail that we need to resolve some of these questions. I'll be using the one we have in Bonn for the next couple months, and I'll do some later vlog entries to show you how all of that works. But for now, we're not going to get into that. One of the downsides to transmission electron microscopy, though, is that we can't do it on living animals. This means that in order to see a process where many steps have to happen in sequence, we have to capture those moments in a series of 2D photographs. Then using the series of 2D images, we can reconstruct what the 4-dimensional process looked like. Ingestion, incorporation, and eventual digestion in Slug takes months, so my boss and I have set up a schedule of time points. We are hoping that the series of photos I make in 2D will show us how this 4-dimensional process happens. I am in the museum tonight because 2 of these time points occur 6 and 12 hours after feeding. When I got to the lab today, I fed some animals that I had previously starved down, and now they have to digest. One animal for 6 hours, the second animal is for 12 hours. The fixation process is really long. It takes about 6 hours for 4 samples, and it's pretty boring, so I'm not going to show it to you here. But if you add that to the 12 hours that I've spent waiting for the animals, you can see why I have to be here all night. Most of the time during the fixation process, I'm just waiting. And the good thing about that is I can work on other projects, like vlog entries. So anyway, thanks for tuning in. I hope you have a great week. Leave comments, subscribe, do all of that kind of stuff, and please keep me entertained. It's gonna be a long night.